The overall goal of this experiment is to investigate the pollinators of Lycoris sanguinea, variety Sanguinea, and their pollination abilities.
This method can help researchers in the pollination ecology field. For example, in decreasing an observer's long-term observation load or focus on a specific pollinator's effect per plant. The main advantage of this technique is that it facilitates the investigation of floral visitor species and frequencies for the analysis of a specific visitor's flower pollination efficiency.
For floral visitor pre-observation, first decide which target individuals in each area will be observed during the same time interval. Next, select 5 to 10 freshly opened flowers or breaking buds as target flowers and begin the visual observation for the period of an entire day, as the flowers can be visited by various pollinators in different time ranges. On the recording sheets, record the species name of each floral visitor and the time of each visitation per hour per flower. Closely observe whether floral visitors touch the anthers and/or stigmata. If they do, record the visitors as pollinators. To capture the floral visitors for their identification and preservation, use a handmade aspirator. Then kill the trapped visitors quickly with ethyl acetate and preserve them in a plastic tube with 100% ethanol. At the end of the observation period, use the morphological features of the visitors to identify the specific names of the specimens.